Hi guys, it's Dr. H here again, and I am here with another review video for you. Today's topic is going to be on mitosis. We're going to talk about the cell splitting up. And I know this is not everybody's favorite topic because splitting up is just so hard to do. It's just so emotional and I just get so torn up about it. It's just so hard. Just give me a moment. Yeah. Okay, I'm good. Don't you dare try to even act like you didn't laugh at that little joke because I know you did. I know you like it. All right, so let's dive on in. Okay, so mitosis. Remember that mitosis is a stage of the cell cycle. Let's just review that little thing. Take 30 seconds here. The cell cycle. What we're trying to do with the cell cycle is we're trying to start with one single cell and we're trying to make a duplicate of it. It's trying to clone itself, whether it be because it needs to repair, needs to grow in the cell body, or what have you. But it's trying to start with one cell and end up with two identical cells. They look exactly the same. But in order to do so, we have to go through the stages. Remember that we go through enter phase first, and then we go through M phase. Remember the enter phase is that stage where you're growing and you're replicating your DNA, and then you're preparing for mitosis. And then your M phase is where you actually start to split up. And remember, the splitting up of the cell happens in two stages. First, we have to split up the nucleus, then we split up the cytoplasm. And when we split up the nucleus, that is called mitosis. But then when we start to split up the cytoplasm, it's called cytokinesis. Got it? Cytokinesis, splitting up the cytoplasm. Easy way to remember it. Yay! Okay. So let's dive on into the mitosis and let's talk about the stages. And remember, the goal here is that we can explain what happens in each stage and then we can identify those stages just by looking at a cell under the microscope and saying, that, my friend, that is a stage of mitosis. That's our goal, okay? Let's stay focused. Okay, so it's first, before we go into that, let's look and see what enter phase looks like. When you look at a cell underneath the microscope, this is what they look like. This over here on the left is an animal cell. This over here on the right is a plant cell. Now this is what your nucleus looks like. You can kind of see that there's DNA in there, but you can't really see those really cool chromosomes. All of those little chromosomes like that we use in class, we said that our chromosomes were like our fingers here. Can't really see them. They're all balled up in a big old ball stuck in here. And you're like, I can't see it, but it's there. Big old ball of yarn. Remember we talked about that. That's in enter phase. You're only going to start to see those chromosomes when you're starting to enter prophase. So let's kind of look at prophase. So here's our sketch. Right now I'll just tell you here is that this video is a compilation of the notes that we actually took in class and also the pictures that I showed in class. So this should look very familiar to you, and you should also have notes that look almost exactly like this in your notebook. Okay, so follow along with me in your notebook if you have them, which you should, and I hope you do. Okay, so first stage, prophase. What's happening in prophase? Remember, those chromosomes have condensed. They're like, hey, I know I've condensed, and I know I need to start splitting up into two daughter cells, but I don't know where I'm supposed to go. Ooh, I'm just going to hang around here. Ooh. That's what's happening in prophase. That's why they need these really cool things called those centrioles and the spindle fibers to come in and help guide them. Remember, like in class, we said that these centrioles are kind of like Spider-Man, and they can like go to the edge of the cell. They go to the poles. The centrioles go to the poles of the cell. There you go. Easy way to remember it. Okay. So the centrioles go to the poles and they're going to shoot out those really cool spindle fibers. Those spindle fibers are going to grab on to those chromosomes and move them around to where they need to be in the cell. Yes, that is the goal here. So this is where you start to see those centrioles and those spindle fibers first start to appear. Those chromosomes are starting to appear. So this is what our sketch looked like. The other cool thing is that you're going to start to see the nuclear membrane starts to break apart or disappear. It's going to disintegrate because when this happens, the whole cell just kind of shuts down and says, hey, we're going to use up the whole entire area of the cell. We're taking it all over. This is our ground now. And so that's where it's going to take up every little space that it can, and then it's going to spread out. So you're going to see that nuclear membrane come apart so that those chromosomes can use all the area that it needs. Okay, so what does this look like under the microscope? I will show you. I'm so glad you asked. Okay, so this is what it looks like under the microscope. You have an animal cell on the left-hand side and a plant cell on the right-hand side. This is what it looks like. Those chromosomes are starting to come out. You can see them, but they're still like, I don't know where to go. Bam, they're all here. You can see that you can actually individually pick out those, kind of like outline all the chromosomes. That's what's going to tell you that it's in prophase. You can see that, and they're not like they're just kind of a disorganized mess. Yes, that's what it's going to look like right here in prophase. Okay, so once those centrioles and spindle fibers can grab a hold of them and start moving them to where they need to go, because they're like, you know what, I got you. I will tell you exactly where you need to go. Just follow me. So that's what happens in the second phase of metaphase. So we've got metaphase. And here's the easy part, meet. This is where chromosomes are going to meet in the middle of the cell. Oh wait, that's a really great way to remember it. Meet. Metaphase, meet in the middle. Those chromosomes are finally going to go from being a crazy hot mess to boom. They're going to land in the middle of the cell and you're going to see them all lined up. Those centrioles totally got them in order, got them in line. Okay, remember that those spindle fibers that were shut out of those centrioles are grabbing onto those centromere. Look at that, we kind of zoomed in right there. Remember those from the last video? Centromeres are holding those sister chromatids, those twin chromatids together. Yes, so we've got them all lined up and now they're ready to split and divide. But what does this look like under the microscope? I think this is kind of the easiest one to pick out under the microscope because this is how cool it looks under a microscope. Look at that. It's like this crazy straight cool line right there. In both cases here, the animal cell and the plant cell. And remember, the colors, they don't actually look like this. We've dyed them. So in order to make the cells look like this, we dye them with certain dyes. So we can just see them and make them more visible. Okay, metaphase. They've met in the middle of a cell. Look at that. Look how cute. They're all lined up like little soldiers. Yay. Okay, so after metaphase, they're lined up. What are they going to do? This is where you're going to go to the third stage. This is where you're going to go to anaphase. This is where those sister chromatids are going to be pulled apart. They're going to be pulled apart to opposite poles. Those spindle fibers are going to retract and pull them apart. Remember this, anaphase, A in the anaphase, and pulled apart or away from each other. A, away. Meta, met, A, away. I'm all full of all these little tricks here, guys. Okay. They're going to be pulled to opposite ends of the cells because it's going to divide in the middle for it. Opposite ends. What does this look like under the microscope? This is so cool. Look at that. You can see them being pulled apart. I love it. Yes, you can see these little rows here being pulled to the opposite ends. Okay, so now that you've got opposite ends, now what do we have to do? Now we're going to condense those chromosomes back down into their two nuclei. This is where you're going to get to your fourth stage, your telophase. This is where you see one whole entire cell, but you have two nuclei. Those chromosomes have been pulled apart and condensed down into two nuclei. They are still one cell because you have not split your cytoplasm yet. You have only split your nucleus. There we go. Pulled apart and condensed back down. Cool. The other big thing that you're going to see here is you're going to see that cleavage furrow come in. This is your little indentation here. This is my awesome drawing. Yeah, it's not that awesome. But you can still see that's our little cleavage furrow. If you can see the cleavage furrow with the two nuclei and there's still some attachment, then you know it's still in telophase. That's going to be your kicker. Okay, so now you got two nuclei, one cell. Okay, now what's it look like under the microscope? Let's look under the microscope. Look at here, look at that. They're starting to pull apart here. You got the nuclei starting to come back together, that nuclear membrane starting to reform, nuclear membrane starting to reform, and you're starting to get that cleavage furrow. See how it kind of curves in? It's like it's gonna like that balloon that we talked about in class. It's pinching off in the middle. That's what's going on here. It's starting to cleave. Same thing over here. It's not completely separated yet, so therefore we're still going to say that it's going to be in telophase. After this, it's going to go into your last stage, which is going to be your cytokinesis. This is where you're actually going to have two identical cells, two daughter cells, completely identical clones of each other, and you're going to see two cells and two nuclei. Those chromosomes have now condensed back into their ball, and so now you can't see them very clearly. It's all just like a big blob, and then there you go. Any questions there? If you have questions, come see me after school. Oh wait, what's that a question? Oh wait, you want to do the mitosis dance? Yes, I know you do. Okay, let's do the mitosis dance. Remember that we can use the mitosis dance with our hands and do it. So here we go. I know you love this. All right, so interphase. Let's show interphase. Interphase, those chromosomes are kind of hidden. You can't really see them. They're just kind of, you know, they're there, but it's all like a big ball. What happens next is in prophase. Prophase, those chromosomes are going to start to come out, and they're like, whoa, I can see here, and I'm here. You can see me, but I don't know where to go. Ooh. And then you got metaphase. Oh, look, they all lined up where they need to go in a very pretty line. Yay. Okay, then in anaphase. Anaphase, they're pulled apart. Pulled apart. You've got them apart on opposite sides of the pole. Oh, like I should probably pump this back a little bit. Okay, opposite sides of the pole. Oh, I guess I need to move over. I'm getting used to this. Okay, opposite sides of the pole. Then what's going to happen in telophase? Those get to start to condense back down into two nuclei, one cell. Again, they're kind of stuck together, so everywhere you move, they're going to move together like a little robot. Yes, we have a robot, telophase, and then we've got, here's the favorite part, cytokinesis. Cytokinesis. Now they're two completely different cells. Ooh, dance it out. Cytokinesis. That's what we call the mitosis dance. All right, so let's do it one more time. One more time. Ready? All right, we've got interphase. Interphase, they're all in a single ball here. Can't really see them. Prophase. Whoa, they're all kind of mashed out. I don't know where to go. Metaphase. Whew. Now we got a line. Anaphase, pulled apart. Telophase, condensed back down, two cells, and two cells, or sorry, not two cells, my computer just kind of started to go into hibernation. Sorry about that. Two nuclei, one cell, and then we have cytokinesis. Woo, cytokinesis, cytokinesis. Yes? Okay, any questions there? If you have questions to come see me after school or before school, come see me in your A-Lab. Come ask me questions. Hit me up on Edmodo. Okay, so study hard. Be awesome. I want to see lots of A's, okay? All right, see you guys later. Bye.